Hello and welcome to this webinar titled Is India Anywhere Closer to Implementing Continuous Bioprocessing, hosted by Biopharma Asia magazine and presented by Dr. Narasimha Rao Nedunuri, CEO of Clonz Biotech, and Mr. Jose Castillo, PhD, CTO and co-founder of Univercells. My name is Stephen Edwards. Now, please allow me to introduce our first presenter, Dr. Narasimha Rao Nedunuri, CEO of Clonz Biotech. Narasimha Rao Nedunuri is one of the co-founding members of Clonz Biotech, a biosimilar monoclonal antibody company based in Genome Valley, Hyderabad, India. He is currently serving the company as a managing director and CEO. Dr. Nedunuri, a molecular biologist turned entrepreneur, has 18 years of experience in the field of life sciences research, including cancer biology, proteomics, and molecular diagnostics. He also had business experience in a USA-based company with the responsibility of establishing a business division for its Indian subsidiary. At Clonz, a 7-year-old startup, along with the co-promoters coming from recognized leaders who launched complex biosimilar MABs, driving the company to emerge as a significant global biosimilar MAB company. I will now be handing over to Dr. Nedunuri. Welcome.
Thank you, Stephen, for the nice introduction. Good morning, good afternoon, good evening, depending on wherever in the world you are attending this webinar. As you know, the topic of today's webinar is India anywhere closer to implementing continuous bioprocessing? So before we proceed to the core point, we will discuss a little bit in detail about what is continuous bioprocessing and what are the key drivers basically, and what are the advantages, disadvantages, enablers, and expectations. So here is our agenda for today's presentation. As I just said, what are the key drivers of continuous bioprocess? That's going to be the first topic, followed by advantages of continuous versus batch. So next is enabling emerging technologies. What are those? We'll discuss briefly about what are those emerging technologies that are enabling this continuous bioprocess. The next topic will be expectations from continuous versus batch processing. And finally, we end up with, is India anywhere closer? Let's discuss about key drivers of this technology. As we all know, it's been much talked about for some time. Is continuous bioprocess technology is another buzzword, or does it have really something significant to offer? Yes, being in this domain for some time, we feel yes, these continuous bioprocess technologies have immense to offer. So if you look at the drivers, key drivers, significant key driver is going to be the processing time, per unit dose. So today we all know that the batch, conventional batch process is a lengthier one, especially in a mammalian-based technology platform. We know it's nearly 20 days where we see a batch coming into a concluding mode and we get the end product. Whereas in continuous bioprocess, we can expect to harvest even daily. So of course it depends on what you want and how you want to set your expectations from the continuous process. So then the second key driver is the the equipment footprint. Of course, even the existing manufacturing has come down significantly with the introduction of single-use bioreactors and disposable-based manufacturing components. But still, the footprint remains higher. Of course, the objective is to bring the footprint as low as possible to control the cost of the goods and keep the cost of the goods low. With the continuous bioprocess, yes, the equipment footprint is significantly lower, and that's going to be one of the key drivers. Then again, the campaigns, the flexibility in duration of manufacturing campaigns. So that's going to be the next key driver. So we know the conventional batches have the campaigns with all SIPs, CIPs, and several other things before we proceed on to the next batch. Even more difficult when we have multi-products, wherein change controls and all have to go in before we get into another product or batch. This continuous bioprocess has some contributions in that domain. We'll see what are those. And so next thing would be how quick we can take a batch. I mean, like rapid response to drug shortages, emergencies, and the supply chain demands. All of a sudden we need a quick material, a drug substance, which is very difficult in a conventional mode to get and generate that material when the need arises. So in these, it's quicker. So the next very important thing that is emerging today is region-specific manufacturing of small quantity biologics. As all of us know, the regulatory is not harmonious today across the globe, and we have region-specific regulatory requirements, and also regions wants to be more independent in manufacturing these drugs in their own territory. So under these conditions, it's extremely difficult to replicate the high volume commercial processes in those territories. So it gives this continuous processes, since the footprint is lower, it gives significant advantage getting into a multiple location based manufacturing strategies. So not only that, we can even have multiple trains. Because it is modular. Sorry for that. The next slide talks about advantages of continuous versus batch. I'm only touching upon the key elements here, not getting in detail about this. If you look at this advantages, as we just briefly discussed in the key driver slide, here modular, being a modular, so and compact, you can have multiple trains versus one single large scale train. So how this is going to help us? Basically, as we discussed in the previous slide, suddenly there is a need to generate more drug substance, or even multi-product facilities. If you have multiple products but the same facility, so these multiple trains can add immense value, making it more modular, making it more ease uh, in terms of it's basically flexible manufacturing. So reduced process scale-up time and complexity is another advantage. So if you see the scale-up time, that's going to be immense when compared, in the conventional mode of manufacturing practices that we have, because the volumes are higher. So the more the volumes we deal with, the more complexities are what we have in terms of the scaling up of activities. Since it is a continuous mode of operation, so that scaling up complexities and the daunting tasks that can be minimized. So next is product quality. Well, this has been a big debate whether we get a better quality in continuous or in the conventional batch mode. But with the advances in process analytic technologies and also steady state operations of this continuous bioprocess, what we have is this is resulting in a higher enhanced product quality. So that's one advantage what we have with the continuous bioprocess. And then being smaller in size and modular, the logistics are simplified. So we know huge volumes, dealing with huge volumes, especially in the classified areas, the highly contained facilities. So it's a challenging affair. So the lesser the volumes what we have, the more comfort and controls we will be having in manufacturing. So when we talk about I'm going back one word to the product quality. So because of these volumes are lesser and the cell viability is more, and the media, so unwanted components that are taken out of the media, so we will have less variants in the process. So these less variants contribute significantly in the product quality. So the next point is stable products can be manufactured with low residence times. So we know some of the substances that get released during the cell culture, especially mammalian based, they are toxic to the cell. We have to keep the residence times as low as possible. So the keeping these residence times low will have much stable expression, and manufacturing efficiency will also go up. It's needless to say that when we control all these things, there is a high productivity due to high cell density and viability. So that's again one more significant advantage what we have with this versus conventional batch processing. But well, do we have, why don't we see them already? So why still we have current technologies going on in the manufacturing of these biologics, especially the monoclonal antibodies production, mammalian-based production. Of course, there is a significant entry in terms of the viral, not vaccine manufacturing, but not already in this. And if you see quickly what is making these current technologies continue to be existing is the stainless steel technology is perceived as a low risk, and disposable technology, what it is there already, it is offering flexibility. Some kind of perfusion technologies are already there, so they are like alternative tangential flow and all. To some extent, they are addressing some of the issues. So still there. And we also have flexible stainless steel technologies, like we have today we have the comfort of disposables in the SS. So comfort of SS stainless steel, but when you are using disposables. So all these technologies that are there have its own advantages. So they are still continuing because if you look at the details, some more details why they are still continuing. Many, I mean, in terms of specifically the batch culture, so many secondary products do not hamper cell growth. So during the upstream process, so if your secondary products are not hampering the cell growth, well, you can, one can opt for batch culture. But this is unlikely for many processes. We see that the secondary products, they do interfere with your product and also the cells. So, and the second reason why they use batch culture is the poor genetic stability of the cell line. Because we know the stability of the cell line depends on what genetics you use. I mean, what genetic elements that you use, they offer the stability. If you have poor genetic stability, if the cell line is not stable enough, getting into a continuous batch, continuous process will be a difficult task. So you are limited to go ahead with a conventional batch culture in that case. So operability and reliability, because it is well established, people are still going with it. So then the economic considerations. Most of the people don't have R&D investments that is going in towards developing these things. So the time and money is a consideration that people are putting across. But today there is a trend, there is a shift, because they are now understanding the advantages of the continuous bioprocess technologies. So waiting a little bit more time and investing a little bit more money, they can be put into a comfortable situation when they get into manufacturing eventually. So there is a serious consideration now. And readily available technologies are one more reason people are still going ahead with the batch culture. But this is also changing. Now we are emerging technologies that are coming up. And one more reason what we can say is here low demand. If the demand is low, then it doesn't make sense to have a kind of fully continuous bioprocess wherein you can use partial continuity in the process and then get away with it. If you see enablers, so we know novel perfusion cell culture technologies that are emerging today. And those are the key, for example, the alternative tangential flow systems, and then we have hollow fiber cartridges, we have micro carriers, like they bind the cells and then do the cell culture. Then, all these technologies, they're helping continuous bioprocess to emerge in a much better way. And we have multi-column chromatography in terms of the downstream processing technologies. So that will significantly change. It's already started changing the biologics manufacturing. So this will contribute immensely in the years to come. Single-use bioreactors, of course, have already penetrated well, and integration of these single-use bioreactors into the mainstream continuous bioprocess is the key here. And we know process analytic technologies, that is going to be one of the key drivers here. You can have real-time monitoring of several parameters that gives more flexibility and great advantage in analyzing the processes. So hybrid technologies, where you only have some processes that are continuous and some non-continuous, and then fully continuous processes. These two technologies are going to be, and how this hardware and technology is basically coming up in these two areas is going to drive basically the fully continuous processes. If you look at different continuous processing technologies available as on today, we can majorly categorize into three of them. One is non-continuous upstream, and then continuous downstream, and then continuous upstream and non-continuous downstream. Then obviously the much preferred mode is the fully integrated and continuous process. So first let us take non-continuous upstream and continuous downstream. This one significantly lowers the downstream footprint. So then that means it is not only the equipment size and number, but also it is the media. It's the media that contributes in reducing the cost and size of operations. But of course, the limitations here are going to be the high upstream process footprint. And we also have some limitations in good continuous downstream process technology, which is still awaited. We have been observing some of the technologies that are emerging, but still they have to be there to fully utilize and make this continuous downstream process very efficiently. Okay, then continuous upstream and continuous non-continuous downstream. If you look at this, we can say it is currently widely used, and then we have ATF systems that are clubbed, or there are several ways people made this upstream process continuous and obtained high cell densities and yields. So that's the advantage what we have today. It's already many people are gaining this advantage by implementing continuous upstream processes. So stable protein production, small footprint of upstream process is one of the key things. But here again, the limitations are high downstream process and footprint. Basically, the footprint of the downstream is bigger. Then the continuous, fully integrated continuous process. Of course, this is the most preferred one, wherein the advantages we can see the smaller downstreams, smaller upstream. Sorry, there's a typo there. Okay. Then the capital exposure is significantly lower. So then we have high productivity. One more advantage here, if we make upstream and the fully integrated continuous process, that's the ability of efficient production of less stable proteins. We know most of the proteins what we deal with in the form of therapeutics are less stable. So the more time they are in the manufacturing process, the more they get damaged. So, and the net yield dips. So we need to ensure that the resident times are low. So here that offers that benefit. But however, this still is a dream that is yet to get materialized. So we have to wait and see how these technologies, basically independently, we have seen these technologies coming up, how efficiently we can bring them together and then make the process fully efficient, fully continuous. We have to wait for technologies to emerge. Well, people talk about the regulatory challenges. Now we know FDA is quite flexible, and objective of everyone is to bring the cost of the goods down and make the drugs affordable. So FDA is, because of this nature of continuous bioprocess technology to bring down the cost, FDA is encouraging these technologies. So that means that they're flexible. As long as you fix on the certain things and make the process robust, so there are no challenges. Some of the challenges we've been talking about is the definition of a lot, of course. Now it's no more an issue because we know we can either fix it at time-based or amount-based. So it's up to one. It's up to the developer what strategy they would like to adopt to fix a batch. Definition of steady state, of course, is one more thing that we need to consider. So this needs to be established well to ensure the product quality. So then process controls that are very critical. So then the engineering solutions. All these aspects need to be looked at to make more regulatory friendly. And then finally, coming to the conclusion where we'll discuss where are we heading. Being in India, closely observing what's happening there, what I can say is some of the technologies are already being implemented, like hybrid process with continuous upstream. That is already there. People started manufacturing biosimilars by using the continuous upstream. What slowly getting in there is a hybrid process with continuous downstream that is now seriously looked at by several players. And finally, we are ready to go into the journey of fully integrated continuous process. So that's where the ambitious journey begins. And people already started investing money in developing fully integrated continuous bioprocess. And if I say that, who are those who are getting in there? Look at quickly, there is surprisingly, it is not a big small molecule pharma. It is not well-established biotech player. It is a startup growing dedicated biotech player that is investing heavily in this. There are reasons for that. The big pharma, small molecule pharma players entering into the biopharma for the first time, they are slow in adopting these technology platforms. Why? Because their preference is always a conventional, well-established process. Reason is simple, is that they lack the biotech exposure, experience, and they want to quickly get into the market. So the second player, the category of player, what we see is a well-established biotech player, which is currently high in their revenues. And then the preference would be for them hybrid mode, especially the upstream continuous process. which is already been implemented. The reason is simple. I mean, they don't have limitations for the capex or space. They're not limited by that. You know, they can build huge facilities because, and also they can capitalize on the existing facility what they have already built because they are already in the commercial operations. That is not forcing them to get in there. And also the quick growth in numbers. So, well, for the biotech startups, dedicated biotech players, it is very important, and they feel that there is a need to get into this domain because of three major reasons. One is the value add. They want to value add the product and build a novel process to stand out from the rest. And the capex limitation is there. They cannot build a huge facilities. We know it attracts huge capital exposure and also operational exposure in terms of the finance. And the limited geography target, because being a small player, limited geography target is one thing that is forcing them to explore these technology platforms very closely. So these, however, I mean, once the technologies, what I believe, what we all believe at Clonz is this continuous bioprocess technologies has immense to offer, and then we are seriously exploring to get in there. So then we have hybrid processes that is already there. So maybe start with that and then slowly move on to fully integrated continuous process. Well, I think that's what I have for today. Thank you very much from the entire team of Clonz, and we'll be glad to take some questions from the audience in the end. Now, I will hand over to Stephen.
Thank you, Dr. Nedunuri. Once again, I'd like to remind our viewers that there will be a live question and answer session at the end of the presentation. Please feel free to send in your questions at any time during the presentation via the questions tab located at the right-hand side of the screen. We will go through these at the end of the presentation. Now, please allow me to introduce our second speaker, Jose Castillo, PhD, CTO, and co-founder of Univercells. After working as a head of viral vaccine industrialization at GSK Vaccines, he started up Artellus with a small group of developers in 2006. At Artellus, he designed and developed bioreactors that transformed industrial operations. With his team, he developed the iCELLis systems and transferred it for more than 50 viral processes over the world. The technology is now marketed by POW, where Jose held the position of Director of Cell Culture Technologies. With the same developers, he started up Univercells in 2013, aiming to develop disruptive manufacturing platforms that significantly impact affordability to vaccines and antibodies. With a background of chemical engineering, Jose's education includes a PhD in applied sciences from the Université de Bruxelles and an entrepreneurship degree from the Solvay Business School. As Univercells' CTO, he is now the architect of highly efficient production processes that will make the cell culture available and affordable for all. The goal is to disrupt the industry and become the leader of comprehensive manufacturing solutions. I will now be handing over to Jose Castillo. Welcome, Dr. Castillo.
Great. Thank you so much, Adam, for this very nice introduction. And actually, thanks a lot, Narasimha, for your very complete presentation of all the concepts behind the continuous manufacturing. So my presentation will be actually just, one implementation of all the concepts that you described. So as Adam was saying, at Univercells, we develop platforms actually for the production of viral vaccines on one hand and for the production of biosimilar antibodies. And here, my presentation will be about the very particular implementation of the continuous processing strategy to a viral vaccine. So as Adam was saying, so our company was started up in 2013. Our goal is to make biologics affordable. We target on 2 applications, viral vaccines, antibodies, and we are developing 2 technologies. Basically, one is what we call Nevoline, which is a concept of what we call a micro-facility approach for the integration of all the unit steps that are needed to produce a biopharmaceutical drug. Can be, as I said, a vaccine or an antibody. And for this to happen, we developed, and we are developing actually, a very special, a very high cell density single-use bioreactor that we call ScaleX. And so the process I will be presenting today is about the production of a new saving IPV vaccine, so polio vaccine. This is a program that was funded by the Melinda Gates Foundation for a total amount of 20 million euros. And the challenge we got from the foundation is to make this possible, to make it possible to produce this trivalent polio vaccine for a total cost of production of 15 cents a dose. Knowing that today, the large companies that produce these vaccines face actually cost of production in the range of $1 to $1.5 per dose. And so we are this year about to finalize this program. The first system will be installed by the end of the year in Europe with the facility, the polio facility of our partner Batavia in the Netherlands. And so I will show you in this presentation, a couple of concepts. First is how do we apply all the rules that Narasimha presented, all the rules related to continuous processing to viral vaccines with a specific case study of the polio vaccine production. And then I'll show you at the end of the presentation what are the main benefits of the new process we have developed. So here, just a couple of benefits that Narasimha already presented. So from our perspective, the main benefits that our partners and customers are looking for when they have to produce a viral vaccine is actually to reduce as much as possible the capital expenditure that is needed to set up a new facility, and obviously also to reduce the cost of manufacturing. The audience probably knows that there is a huge pressure on price for vaccines, especially through the programs of the immunization programs funded by UNICEF. So cost is of huge importance for viral vaccine manufacturing, and everything is related here, or everything is focused here on reduction of footprint. So having small equipment, no whole tanks, and optimal use of utilities. Then something that Narasimha also mentioned is, the time you need to build a facility. So especially in cases of outbreaks, emergencies, and at the end of the day, the product is product safety and consistency and product quality is obviously of high importance, and this is something that's a benefit that can be brought by the manufacturing approach we are developing. In terms of adoption, Narasimha also mentioned, so who is on continuous? So today, especially, in recombinant protein manufacturing, so we all know that Amgen is putting lots of efforts, lots of energy, lots of money on the table to make this a reality, and a couple of other corporations like Genzyme, Bayer, J&J, and so on. And today, I think that there is no excuse, if I might say, because the regulators are actually very, very open to new technologies. The only thing they want is the manufacturer to provide the evidence that the product is safe, consistent, and efficient. Now, if I go into, the challenge of implementing continuous bioprocessing for viral vaccine, the main challenge for viral vaccine manufacturing is between 1 day and up to, let's say, 20 days after the virus infection, the virus kills the cells. So there is no host cell anymore to continue the production. So the strict implementation of continuous bioprocessing is just not possible. And so here our challenge was actually to balance the risks and the benefits. So on the risk side, most of the partners or customers we are talking to, they are really afraid of changing the cell substrate or the virus strains. So that was a given in our development, is that a vaccine manufacturer that has a vaccine on the market and who is interested in improving the manufacturing of its vaccines doesn't want at all to change the substrate or the virus strain. So this is a given. And so, as I said, the virus will kill the cells. So our mission was actually to find a way to implement or to have all the benefits of continuous bioprocessing, knowing that the virus will kill the cells without changing the cells and without changing the virus strain. So actually what we did here and what I will be presenting is actually the way we tuned or we changed the process to make it continuous during the period of time available between the virus infection and the virus harvest and purification to avoid all the intermediate tanks, to avoid the large volumes, and then to benefit from the main benefits actually of continuous manufacturing. And so this is what you will see in the next couple of slides. So here is actually the principle behind our manufacturing platform. So everything we do is based on what we call intensification and chaining. This is also something Narasimha mentioned. So to reduce the footprint, there is only one way, it's to intensify the process. So to dramatically increase cell density, and you will see the way we want to achieve this intensification. So this goes through the new ScaleX bioreactor we invented. And then once you succeed with the intensification to grow, in the case of vaccines, adherent cells that are the hosts that will be used for the virus to duplicate. So once you succeed with this implementation of very high cell density, you obviously need perfusion. And so the perfusion is here leveraged because after the virus infection, the virus is released into the perfusate of the bioreactor. And so the only thing we did here is to couple this perfusion flow rate together with all the downstream steps that are needed to purify a purified vaccine. So the clarification, benzoate treatment, clarification, concentration, acidification, and purification by chromatography. And so here, this is what we did. So all these steps are chained, so coupled with the perfusion of the high cell density bioreactor to dramatically reduce the size of the equipment and the size of the facility. So if I show you a couple of slides on the way it will look like, what you see here on the left-hand side of the slide is actually what we call a microfacility. So the Nevoline system is actually made, in the case of polio, actually of 3 cabinets. Cabinet number 1 is used to grow the cells at high cell density using the ScaleX bioreactor and the perfusion. It's also used to grow the virus after the cells populate the available space in the bioreactor. So this is what I would call the upstream cabinet. In the case of polio, for containment reasons, this cabinet is actually an isolator. So it's operated at negative pressure, and so there is no direct contact between the process space into the cabinet and the operators. So this is to comply with the very strict regulations of WHO for the manufacturing of polio. So after we grow the cells, we produce the virus in the cabinet number 1. We go to the cabinet number 2, in which we implement the purification process. And so as I said, purification process is essentially DNA digestion, clarification, and bi-dilute chromatography. So we have a very small volume that goes out of the upstream process at very high cell density, reason why we were able to dramatically reduce the size of all the DSP steps. And after the virus is purified in the purification cabinet, again, in the case of polio, it moves to the 3rd cabinet, in which we implement the inactivation to make it an inactivated polio vaccine. So here, actually, again, this is the consequence of the approach. So we use, as I said in the beginning, existing standard, well-characterized WHO zero cell lines, attenuated sabin polio strains, very well characterized and accepted by the regulatory authorities, the ScaleX high cell density bioreactor in perfusion, so that provides the intensification we need to implement the chaining of cell culture and virus production process. So in essence, this is the reason why we call this a microfacility. So all the production steps happen into this very small series of 3 cabinets that are isolated in the case of polio. So obviously I won't go into the details of the drawing you see here, but actually that was, from the engineering perspective, that was our approach, is actually to design all the steps that are needed, since the installation of the single-use Calyx bioreactor through all the process until the dismantling of all the single-use components and actually the dismantling or the removal of the purified drug substance. And so all this, and it seems that I lost my slides. I don't know, Adam, if you can help me with that. I don't know if you see them.
No problem. Give me 1 second. Do you want to quickly let me know what slide you want?
Actually, it was the slide with the flow chart, with all the inlets and outlets of the process. But actually, as I don't see it anymore, I cannot tell you what's the slide number actually.
Was it this one?
No, I don't see it. Maybe what I will do is let me check the internet connection. Maybe Adam, I will ask you to go through the slides. I will use the
No problem. We can start from is it the engineer drawings?
Exactly, engineering drawings.
That is the one that's up. So if you go through it, let me know and I will carry on. Sorry for the technical difficulties.
Yeah. So I will maybe, to make things easier, I will use the presentation on my computer and maybe ask you to go through the slides. I will just tell you the slide number because I don't see anything here anymore.
No problem.
Okay, so I opened it on my computer.
Yep.
Done, and I go to the slide number 10, which is actually the engineering drawings. Is that the one you have, you are presenting to the audience, Adam? Okay, great. So I was just saying that, yeah, so this is what we did from the engineering perspective, is actually to, to go through, all the detailed steps of the process. So from all the inputs and outputs of the bioreactor, of the virus concentration system, the DNA digestion, clarification, binding, and so all the steps. And then if you go to the next slide, Adam, please, slide 11. So then all the steps were designed around the high cell density bioreactor system, so the ScaleX, and then we specifically designed all the manifolds that are needed to implement all the operations, and they were all optimized to be as small as possible to be able to implement them into a very small cavity, which is actually, as I was saying, in the case of polio, an isolated cabinet. In the case of other vaccines, it could be a biosafety class 2 cabinet, for example, for many of the viral vaccines that are today commercially available. And so, based on this detailed analysis of all the steps of the process, we designed actually the 3 cabinets that we see in the slide 11. So the one related to upstream operations, downstream, so clarification, purification, and then dilution and inactivation. And as you can imagine, this has a dramatic impact on footprint. So the total footprint of all the operations at large scale, because 1 unit of this will produce 500,000 doses of the polio vaccine per run. So that the total footprint is below 7 square meters. So that has a dramatic impact on footprint and consequently on capital expenditure. So if we go to the slide 12, so the next slide, here we can see that, from the process reproducibility perspective, so process robustness, after 18 months of development, today we are there. So what you see here on the left-hand side of the slide, that's in average, we superpose almost, I think, yes, 40 runs of cell growth into the high cell density bioreactors, where we achieve in the range of 40 million cells per milliliter of bioreactor operated under perfusion. Next figure is actually the kinetics of the production or duplication of the virus. So in the case of our polio, as the audience might know, there is 3 types of polio that are part of the vaccine. The 2 most important from the production perspective are the polio type 3 and the polio type 2. And so for these 2 types, we were able to increase by 40-fold the volumetric productivity for type 3, 25-fold for type 2. So what does it mean? It means that the bioreactor size can be decreased by 25 to 40-fold compared to a reference process that uses batch production into classical stainless steel or single-use agitated bioreactors. And this was actually the main goal, to reduce the size of the bioreactor, for the bioreactor to be implemented into a very small cabinet. And then one of the consequences of this perfusion-based process is actually that the harvest we get is cleaner when compared to a reference process. At the same time, we developed quite an improved DSP process as well, for which the antigen recovery is actually 90%, which is record in the field of vaccine purification. And so actually with this process in hand, we can produce up to 2.5 million doses of type 3, monovalent, 1.5 million doses of the monovalent type 2, which when you apply the final inactivation and formulation of the 3 types, gives you a little bit more than 500,000 doses of the trivalent vaccine per batch in a very, very small system that you can see in the next slide, the slide 13. So this is the actual engineering drawings of the complete process into 3 isolated cabinets. On the left-hand side, so you see the bioreactor, the harvest tank, 50 liters, very small tank, where we harvest a highly concentrated virus that moves to the cabinet number 2, where you can see the filtration systems, as well as the chromatography, which again is operated in sequential continuous mode. And after purification, the cabinet number 2, we go to the cabinet number 3 for the implementation of the inactivation. And that's actually the system that is currently produced at Univercells, and that will be used in the Netherlands with into the polio facility of our partners of Batavia Biosciences for the final scale-up of the process and the production of clinical material that will happen next year. And so if you go, Adam, to the slide 15, we can see here what really matters for our partners and customers. So at the end of the day, everybody is interested in great technologies, but the main benefit for them is how much does it cost to produce the vaccine, should I succeed with the implementation of this technology. And so today, based on the yields we have, on the process we have developed at small scale, this is the extrapolation of what the cost per dose, total cost of production will be. So we are today in the range of 22 cents per dose, based on the final optimization of the process we are implementing. Knowing that, and this is what we summarize in the next slide, the slide 16, knowing that with the reference process that a couple of companies use to produce IPV, the total cost per dose is more in the range of, above $1 per dose. Today, we will be at 22 cents, and that has a real impact on the sales price. Knowing that today, UNICEF is willing to buy the standalone polio vaccine at $1 per dose. So obviously, with this new process, it makes it possible to sell it at $1 and to build up a sustainable business. So this is really of high importance for our partners. that the final cost per dose is achievable to make it a sustainable business and to make it affordable. And at the same time, an important number you see on this slide is the expected capital expenditure to set up a production facility, which, based on the cost modeling we developed at Univercells in Batavia, should be in the range of 15 to 20 million for an up and running facility that would produce 40 million doses per year, to be compared to a traditional facility that would cost between $200 and $300 million, so 10 times more, to produce indeed 16 million doses instead of 40. But here you see, the main benefit that our partners are expecting is this: high quality, very large outputs, dramatically reduced capex, and significantly reduced cost of goods per dose. Now, if I go to the to my conclusion in the slide 18. So actually, this is again, and this is one of the applications of the continuous manufacturing that Narasimha presented more broadly. But in the case of viral vaccines, today, what the virus vaccine producers are expecting is a reduction in capital expenditure, reduction of cost of goods, and obviously footprint, high containment, safety, and flexibility. This is something that Narasimha also mentioned. So this platform should be applicable to many viral vaccines, even if we developed it for the production of polio. So that being said, I thank you everybody for your attention. I'm sorry for the technical issue, and I wanted obviously to thank the Gates Foundation for the great support for this program, and our partners of Batavia in the Netherlands, and Millipore Sigma that actually provide the great purification technology we are using for this program. So thank you so much.
Thank you, Dr. Castillo. Now we will begin the question and answer segment of the webinar, and once again I'd like to remind the audience that you can still send your questions via the questions tab on the right hand side of your screen. Now we have one question here for Dr. Nedunuri. One, how can you justify the use of high volume of media, which is an expensive component leading to high costs of goods?
Okay. So the media, yes, of course, the media requirement is higher, but when you attribute the media requirement to per gram product, it is not significantly higher, because we know the volumetric productivity is high in continuous process. So that media requirement per gram is slightly higher, not much higher, but the other components that are there in this, the cost components, they contribute significantly towards the low cost of goods. So that gets nullified. Hope I answered the question.
Thank you. And our next question is for Dr. Nedunuri again. Which one is of high priority, whether continuous upstream or continuous downstream?
Well, of course, both of them, ideally. There's no such preference. It depends on your product and what are you looking for. If the product is quite unstable, you may go with a continuous upstream, which becomes mandatory. Then if the product is stable, irrespective of whether the product is stable or not, if other parameters you consider, basically the continuous upstream contributes high product quality, and the downstream, apart from high product quality, it offers the low cost of goods, so, and ease of operations. So basically the product quality for upstream, better cost of goods and quality for downstream. So it's up to you. I mean, so the obvious answer is both of them.
Okay, thank you. We have another question for Jose Castillo. The process you have presented for virus production is not really a continuous process. So how can you explain the benefit it brings in terms of capex and COGs?
Actually, this is what I tried to explain during the presentation, is that indeed when you spike a host cell with a virus, at the end of the day, the virus will kill the cell. So indeed, you cannot imagine a kind of continuous process that would run over 2 months, like you can develop for CHO cells in suspension. So this is clear, this is true. So here what we did is to try to develop a process that would use the time window we have access to between the virus spiking and the cell death, actually to implement all the rules of continuous manufacturing during that period of time, which means that we were able to kill, all the major components of cost, like whole tanks that has an impact on footprint, and all the large volumes needed for the purification. So at the end of the day, we have a batch, this is true. So when you start a run, at the end of the day, you have one batch, but during the execution of that batch, we applied all the rules of continuous manufacturing. And that's the reason why we get the same benefits.
Thank you. And we've got time for questions. Can the process and related equipment you have presented be used for other applications like viral vectors? I think this one was for you as well, Jose Castillo.
Oh, yes, indeed. So actually, this was one of my conclusions, is that everything here was developed for a specific application first, which is polio. But at the end of the day, what we have developed is a high salinity bioreactor that can grow cells to a very high concentration. We developed a perfusion process. That perfusion process is coupled with a concentration system, clarification system, and purification system. So at the end of the day, if you change the zero cells and you use MRC5 cells, X293 cells, BHK cells, or any other cells, after growth, you infect these cells to produce a virus, actually the process would be very, very similar. So yes, indeed, it is applicable for viral vectors, certainly.
Thank you. This next one is open to both of our presenters. While thinking about a continuous process, how much flexibility does the operator has in an ongoing process?
Well, Jose, should I take, or you're going to take?
Oh yeah, go ahead,
okay.
Narasimha.
Okay. The continuous process, if you talk about the flexibility, yes, it has it offers a lot of flexibility. For example, if you look at from a transition from upstream to downstream, there are several whole tanks. Or from in between the downstream processes, you have whole tanks and transfer mechanisms. Several of them can be reduced or even make zero. So that makes life a lot easier. So it's being even the size being smaller. It's like it's modular, we can say, and doing the tech transfer from one side to the other gets significantly easier. I think there are several ways that this becomes flexible. I think these being key flexible factors. Answer that.
Thank you for that. Before I finish the webinar, I'd like to ask our presenters, Dr. Narasimha Rao Nedunuri and Dr. Jose Castillo, if they have any closing remarks.
Well, the continuous bioprocess is evolving, and the need is much higher, especially when the more number of players, when there is a need to reduce the cost. So we need more technologies that to come up rapidly and bridge that gap. And we hope these emerging technologies to be adopted as quickly as possible to make the drugs affordable. And with that, I conclude saying thanks to the organizers and thank you, audience. This is all from Team Clonz.
And nothing to add to that. So thank you so much for the organizers and to the audience and for the organization of this webinar.
Thank you, Burke. I'd like to take this opportunity to thank both of our presenters, for Dr. Nedunuri and Jose Castillo, for sharing their knowledge with us. I would also like to remind our audience that they can view this webinar on demand by visiting biopharmaasia.com. Thanks once again to our presenters. Thank you, everyone, and have a great day.